Hello, this is a quick revision video for Unit B4 from the AQA GCSE on the different ways that plants use the glucose produced in photosynthesis. So we're going to do this as a concept map. We will start with glucose, and one of the uses of glucose is as a reactant for respiration. Could be aerobic respiration, could be anaerobic respiration. And what respiration is going to do is release energy. That energy can be used for active transport, moving substances against a concentration gradient, and can also be used to build molecules. That active transport is going to uptake things like nitrates from the soil, so that's active transport of the nitrates from the soil using energy because it's moving it against a concentration gradient, and those nitrates are going to be used to build amino acids. It's actually nitrates and glucose together that are going to do that. So glucose is used to build molecules such as amino acids using nitrates from the soil. There are other molecules that are being built from glucose as a sort of starting point. One of them is cellulose. Another is starch. And the last one we're going to talk about is lipids, or are lipids. We can go through the function of these larger molecules built using glucose. We'll start with the amino acids. Amino acids are joined together in a process called protein synthesis at the ribosome to make proteins. Cellulose is used to make cell walls. Starch is essentially a whole bunch of glucose molecules put together, so it's storage of glucose. And lipids are another way that plants can store energy. They're also used for making things like cell membranes. In terms of B4, that's all we need to remember for the uses of glucose, but we can take this opportunity to do a little bit of B2 revision, specifically the food tests. We'll start with glucose. Glucose is a reducing sugar, which means we can test for it using the Benedict's test. And in the Benedict's test, it starts out blue, and in the presence of a reducing sugar like glucose, it'll turn green and then orange and then red. If it turns green, that means there's a little bit of reducing sugar there, a little bit more, a little bit more, a little bit more, until lots of reducing sugar means it turns red. Moving on to starch. For starch, we're going to use iodine. And iodine starts out sort of browny orange and will turn blue-black. Moving on to lipids. For the lipids, we're going to do the Sudan 3 test. And what you end up with is a layer of red at the top. So for example, you might have a boiling tube that looks like this, and what you'll have is the lipids separating out at the top from the water and a red layer appearing. For proteins, we're going to be using the Biarritz test. And that starts blue, turning to purple if there is proteins there. Okay. Hope this has been useful for you. We do need to know the uses of glucose for Unit B4, and then we've also thrown in a little bit of food testing revision from B6. Thank you very much. Bye-bye.